Shall I start, sir?
Yes, please.
A very good evening to all the participants. Let me start with some words. The beginning of knowledge is the discovery of something we do not understand. With this, I, Shreya Mishra, the co-organizing secretary of 1 month international training on precision agriculture, organized by CSJM University, Kanpur, and AgriMeet Foundation in collaboration with
organized by CSJM University, Kanpur, and AgriMeet Foundation
in collaboration with ICRISAT Hyderabad, welcomes you all to day 19th of the International Training on Precision Agriculture. The title of today's training is
National Training on Precision.
Advanced
मैम
Post.
आपका एक सेकंड थोड़ी सी ग्लिच आ रही है. आप क्या मोबाइल पे भी ऑन किए हैं क्या?
No, sir.
आपकी तरफ से डबल वॉइस आ रही है और वीडियो बार बार क्रैश हो रहा है. हाँ, आप प्लीज कंटिन्यू करें अब.
Sir, shall I?
हाँ, यस प्लीज.
Sir, should I start with the beginning?
नो 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 नो, जहां से आप कर रहे थे वहीं से करिए.
Hello.
आप जहां से कर रहे थे वहीं से प्रोसीड करिए.
So let me start. Organizing secretary of 1 month international on precision agriculture, organized by CSJM University, Kanpur, and AgriMeet Foundation in collaboration with ICRISAT Hyderabad, welcomes you all to day 19th of the International Training on Precision Agriculture. The title of today's training is Advanced Post-Harvest Technology for Accelerated Crop Improvement. To share the views on the topic today, we have an eminent speaker, Dr. Tharanya Murugushan. Let me take the honor to it is Dr. Tharanya Murugushan is the DST SERB National Postdoctoral Fellow, working on low and crop production related traits, water saving trait for drought adaptation in pearl millet through physiological approaches. She has worked in different phenotyping platforms such as LizzieScan and Lysimeter. She has approaches and 3 research articles and 2 book chapters. Further research interests include rain quality and water stress conditions. Ma'am.
Thank you for your introduction. Let me share my screen first. Is it okay if I share now?
Please share your screen, madam.
Okay. Can someone confirm whether my screen is visible?
Yes, ma'am.
Hello,
Completely
ma'am. Are you there in the session?
Yes.
हाँ हाँ ठीक है, हो गया श्रेया जी, हो गया, विजिबल है. ठीक है मैम, प्लीज आप प्रोसीड करिए.
Okay. Hello everyone. Good evening. I'm Taranya. I hope you are having a pleasant evening. The topic that I will be presenting today is the advanced post-harvest technology for accelerated crop improvement. Here is a small overview. In this presentation, we will focus on the importance of technology-aided post-harvest process. Specifically, we will explore the technologies implemented at ICRISAT to enhance the post-harvest activities. So we have 3 different technologies: X-ray computer tomography, X-ray fluorescence, and field data collector, which is the harvest master. Each of these technologies plays a very crucial role in the post-harvest operations. First, let us know why we need technology and sensors for post-harvest activities. Well, we all know that doing post-harvest activities manually requires a lot of labor, money, as well as time. Also, for the wet lab work, we use a lot of chemicals that are not good for the environment and human as well. That's why we have adapted technologies that can help us to do these activities more easily and efficiently in an environmental friendly way too. These technologies are non-invasive, cost-effective, and require less manpower. Using sensors-based phenotyping, we can examine the fodder and grains for organic components, inorganic components, and physical properties as well. For looking at the organic components, we use NERS machine. For inorganic composition of iron, zinc, and calcium, we use XRF. For physical properties of grains, we use X-ray computed tomography. And for harvest processing related measurements, we use Harvest Master. I hope you already heard about the NERS in the previous presentations by Dr. Kritika Anbalagan. So today we will look at the other remaining sensors available at ICRISAT for post-harvest operations that we use for our research purpose on a routine basis. First, let's look at the X-ray computed tomography technology. CT scanning is commonly used in medical imaging to visualize the internal organs and tissues of humans. In agriculture, CT usage refers to the use of X-ray imaging techniques to study the internal physical structures of plants. And this allows a non-destructive assessment of plants. For example, in breeding programs, after the harvest of groundnuts, all individual groundnut pods needs to be open, which is usually thousands in numbers, to get the kernels for measuring traits like yield. This takes a lot of time and it requires many labors too. To speed up this process, we have CT technology. With CT scanning, we can quickly scan the pods even without opening them and get the necessary data in just few minutes. This means we can complete the post-harvest work much faster, and the data we collect is more accurate, avoiding any mistake that can happen while doing manually. The principle behind this technology is very simple. The sample is placed between the X-ray source and a detector. When the X-ray pass through the sample, some of them are absorbed while others are emitted. The emitted X-rays are then detected and displayed as an image on the computer screen. To understand and interpret these images, special algorithms and softwares are used. These algorithms and softwares process the image and provide us with the necessary data or information about the sample. Let's look at a case study we did here at ICRISAT in Groundnut using CT technology. We are working together with the Groundnut breeding team to accelerate the post-harvest activities of Groundnut. In this case, we are specifically interested in very crucial traits like kernel size and shelling percentage. To explain you what is shelling percentage, it refers to the amount of outer shell or husk that is removed from a crop like groundnut. It is the measurement that tells us how much of the shell is taken off to reveal the edible part, which is the kernel here. So a higher shelling percentage means more of the shell is removed, resulting in a larger portion of the usable kernel. It is an important trait to consider in groundnut breeding as it affects the overall yield and quality of the crop. This is the sample holder that we use for arranging the groundnut pods, and it is made from a special polystyrene material. So after placing those pods within the scanner, this is how the image of the CT scan will look, and this is the image of the CT scan setting we use to scan the groundnut pods. This we have optimized previously for groundnut. In addition, we have crop specific settings for different types of crops. This is how the CT image appears after the groundnut scanning. Here we can see there are variations in kernel size that vary from extremely small to larger size. The beauty of this technology is it allows us to detect all these variations in kernel size in a non-invasive way, even without opening the pods. Once the image process is complete, the algorithm takes over the analysis of the images. It is designed to identify and separate the different components such as the pods, kernels, soil, and impurities. The topmost left image that you see is the raw image obtained after the CT scan. Using this image as an input, the algorithm works to detect the borders of the shells, and then it outlines both the shells and the kernels, what you see in the next images. And it identifies the other parameters like dirt, foam that we use as a sample holder, the kernel threshold, and then the shell threshold. Also, the algorithm uses a reference value to aid its calculations and estimations. So the data that we receive after all this image processing is the shell weight, kernel weight, and kernel count. Based on this data we receive, we further calculate the shelling percentage and 100 kernel weight that gives an idea about the kernel size. As part of our recent project, we performed CT scans on a large number of groundnut samples, close to thousands in number. Here is one of the results that shows a very clear variation in prides that are shelling percentage and kernel size. In the image obtained from the CT scan, we can visually observe that the shelling percentage and kernel size differ significantly across the sample groundnuts. This variation indicates that different groundnut genotype has different characteristics in terms of shell removal and kernel size. To validate these findings, we consulted with the groundnut breeding team who has extensive knowledge and expertise in groundnut genetics and traits. They have verified the range of values obtained for both shelling percentage and kernel size that aligns with what is typically expected and observed in groundnut populations. In other words, the variations we see in the CT scan image are consistent with the naturally occurring diversity present in the groundnut crops. These results are valuable for groundnut breeding program as they provide insights into the genetic variability within the sample groundnut population. By understanding and characterizing such variations, breeders can make informed decisions to select and develop groundnut varieties with desired shelling percentage and kernel size. Additionally, we also conducted a comparison between the ground truth data, which is the actual measurements taken manually, and the values predicted by the CT scanner. Remarkably, we noted that CT predicted values for both shelling percentage and kernel size are closely aligned with the ground truth data. This indicates that the CT technology we use is reliable and accurate in estimating these traits. Our team has recently published these findings on this computer tomography. And if you are interested in learning more, you can access and read this publication online. This publication provides you comprehensive details about the research methodology that we used, the results that we got, and the significance of the findings in the field of groundnut breeding. Apart from groundnut, CT has lots of scope to explore other crops and traits as well. In this particular example, we have images of foxtail millet panicles after they were harvested. What is very interesting here is, if we look at the red box, where we can see pictures of the panicles taken with the mobile camera, all these 3 panicles look quite similar. However, when we examine the CT images of those panicles, what we have in the left side, we notice some important differences. So the top image shows you a mature and healthy panicle. The bottom one has very poor rind filling, and the middle one exhibits signs of infections or disease, which is very beautiful. Similarly, I have another image here for you, and in this case, it is wheat. Again, the pictures within the red box, if you look at it, which is again captured with a mobile camera, appear to be very similar, both of the images within the red box. However, the CT image helps us to differentiate the level of grain filling in both the panicles of wheat. The CT image that you see at the bottom has a good grain filling, and the image at the top shows a poor grain filling. And this is visible only through the CT scan. In addition, we also use CT technology to examine rice for specific qualities like chalkiness, chaffiness, and head rice recovery. This helps us to determine if the rice grains are suitable for eating and processing. Moreover, we plan to expand our research with the CT to include seed viability testing. This testing is important for gene banks because it can help us to judge whether the seeds that are stored can still grow into new plants. It ensures whether the seeds that are preserved for the long time can be used in future crop breeding process. In addition, we also wanted to use the CT to study the dal recovery from pigeon pea. This way, we can screen a large panels for identifying best pigeon pea variety for dal recovery. CT technology is not limited just to these crops or traits. We believe it can be used for many other crops and traits as well. It's all the matter of having the specific algorithm in place, which can be developed with the help of IT people. And this CT can help us to speed up the work done after harvesting crops in different breeding programs. By using CT scans, we can collect data faster, enhance accuracy, and quickly analyze the very crucial traits for improving the crop research. Shall we move on to the next technology? Okay, the next is the X-ray fluorescence. While the computer tomography machines can examine the physical properties of grains, XRF machines are specifically useful for determining the inorganic content of grains such as iron, zinc, and calcium. XRF technology employs X-rays to stimulate the atoms within the sample, causing them to emit distinct fluorescent X-rays. These emitted X-rays are then measured and analyzed to quantify the concentration of iron, zinc, or calcium. The utilization of XRF analysis offers several advantages for assessing iron, zinc, or calcium content. It is a rapid, non-destructive method that requires minimal sample preparation, making it suitable for grains, soils, and plant tissues. Moreover, you can avoid the usage of chemicals, which is usually used in the wet lab work. Also, the results obtained are quantitative, providing precise measurements of iron, zinc, or calcium concentration. Both humans and plants require iron and zinc as essential micronutrients. By measuring the content of these micronutrients in agricultural samples, we can evaluate the nutritional value of crops and monitor for nutritional deficiencies. This understanding of micronutrient levels enable us to improve crop nutrition and make informed decisions to enhance both the plant and human health. XRF instruments are user-friendly and require minimal training, making them widely used in agriculture for quality control and research purpose. Principle: X-ray fluorescence works by using special rays to interact with the objects. When an object is exposed to these high energy X-rays, the atoms inside it absorb the energy and some of their electrons get knocked out. This creates empty spaces that needs to be filled. So other electrons in outer shells move down to fill those empty spaces. When they do that, they release extra energy in the form of fluorescent X-rays. These X-rays have different energy depending on the elements present in that object. By measuring the energy and the amount of these emitted X-rays, we can identify the elements present in that sample and measure the concentrations. Each element produces unique fluorescent X-ray energies, so we can detect and measure multiple elements at the same time. The XRF machine has three important parts, and an X-ray source that sends out strong X-rays, a place to put the sample, and a detector that captures the fluorescent X-rays released by the sample. When we expose the sample to the X-rays, it absorbs the energy and emits fluorescent X-rays. The detector collects these fluorescent X-rays and changes them into electrical signals. These signals are then processed to create a graph that shows what elements are present in that sample. To accurately measure the amount of iron, zinc, or calcium in a sample, the XRF machine is set up using known samples or calibration curves. This calibration process helps convert the measured X-ray strength into specific measurements of iron, zinc, or calcium in that sample. So for this let's look at a case study. We conducted a case study on pearl millet diversity panel, focusing on the estimation of iron content using XRF. The results revealed a significant variation in iron content among the different genotypes of pearl millet, and interestingly, some of them even had higher levels of iron content than the existing cultivars that are shown in blue. So this is really important for plant breeders because it gives them a valuable information that can help them to make better decision when selecting pearl millet genotypes for their use in breeding process. Similarly, we also assessed zinc in pearl millet, and there too we had a significant genotypic variation for zinc content across the pearl millet diversity panel. And in that too, some of the entries within the pearl millet diversity panel had higher levels of zinc content than the existing cultivars. Furthermore, we check the accuracy of XRF measurements by comparing them to the actual observed data. For example, when we estimated the amount of calcium, we found a strong connection between the XRF measurements and the actual values. If any one of you is interested in knowing more about the XRF, please refer to this article that is displayed in the slide. This is by Dr. Govindaraj Mahalingam. It is one of the very interesting paper that can provide you with the basics of XRF. Let's move on to the next sensor. We have here is the field data collector, and the brand that we use is Harvest Master. Why we need Harvest Master? Because it's mainly used to collect real-time data quickly and accurately right in the field during the harvest process itself. One of the key features of this sensor is its ability to collect data immediately after the harvest with great precision. Harvest Master is very helpful in collecting information about grain weight, grain moisture, and test weight, which will tell you about the grain size. These factors are important in evaluating the quality and market value of harvested grains. By using Harvest Master, farmers and researchers can gather precise, up-to-date data, which can help them to make better decisions and improve the post-harvest operations. The Harvest Master machine is usually combined with the harvesting machine to instantly measure grain weight and moisture content. This data is crucial for several reasons. Moreover, the Harvest Master offers high throughput processing with each sample in just 30 seconds or a minute in the field while maintaining high accuracy. For farmers, knowing the moisture content is very important for determining the optimal conditions for drying and storing harvested crops. By implementing appropriate drying and preservation methods, they can maintain the quality of these crops and minimize loss. Excessive moisture can lead to mold growth during storage, while low moisture levels can negatively affect germination. Therefore, farmers can make well-informed decisions about drying and storage based on the data provided by the Harvest Master. For researchers, moisture levels in the field can be heterogeneous for several reasons, causing variations in moisture content across different plots. As a result, the grain weight harvested from each of the plots needs to be adjusted based on the moisture prevailing in that particular plots. The Harvest Master is particularly useful for this purpose. By using Harvest Master, researchers can achieve precise results and make informed decisions about the performance of different genotypes in terms of yield from the field. To provide you an overview of its functioning, the Harvest Master operates in the following manner. First, the harvested grains are loaded into the weight hoppers. Then the sample gate is opened to transfer the grains into the test chambers. Within the test chambers, the sample weight, moisture content, and test weight are measured. Finally, once the measurements are complete, the samples are removed from the test chambers. And all these process gets completed within a minute. Apart from the existing inbuilt equation that are in Harvest Master, we have also developed equations for different crops, making it more precise, reliable, and crop specific. We also carried out a cross-validation process to validate the accuracy of our Harvest Master measurements. This involved comparing the measurements obtained from the Harvest Master with the ground truth data that we generated manually. An example here shown is the assessment of moisture content, where we identified a significant correlation between the measurements obtained from Harvest Master and the actual values. This shows that Harvest Master is reliable and precise in accurately estimating the moisture content. So, in summary, the utilization of advanced technologies and sensors in post-harvest activities offers numerous benefits. The use of X-ray computed tomography technology, X-ray fluorescence, and Harvest Master enables data collection and analysis for improved decision-making. X-ray computed tomography provides valuable insights into internal structures and quality of harvested agricultural products. X-ray fluorescence allows for elemental analysis, providing valuable information about the composition of agricultural commodities. Harvest Master facilitates on-the-spot data collection, particularly for crucial measurements like grain weight, grain moisture, and test weight. By leveraging all these technologies, post-harvest operation can be optimized, leading to enhanced efficiency and productivity. Thank you for all your attention. Let me know if you have any question based on this presentation.
रोहित जी प्लीज चैट बॉक्स स्टार्ट करिए.
Okay, sir.
मैम आप प्लीज चैट बॉक्स देखिए, चैट बॉक्स.
Okay. CT scan can be useful for crops whose biological and economic yields are same. What can be the post-harvest factors to be considered for them? Richa, could you please rephrase your question? Okay, let me move on to the next question. How can you decide seed viability by using CT scan technology? Okay, thank you for this question. So using CT mission, how we can decide the seed viability? So what happens when we store the seed samples for a longer time? The viability can be lost in two different ways. One can be due to the biological reasons, and then the other could be due to some breakage or some physical distortion of the seeds internally. So if the viability has been lost biologically, this can be sorted using a nurse. So we can identify using nurse machine. But then if the viability loss is due to the change in internal structure, let's say some breakage inside the seeds, then it is easily visible through the CT machine. And this is how we can use the CT machine for testing the seed viability. Let me move on to the next question. So, what are the methods opted for calcium and micronutrient analysis in pearl millet? So at ICRISAT, we use this XRF machines, we use the XRF machines here. And how is XRF and CT is used for horticultural crops? Well, whatever crop it is, it all depends what we need to research out of these crops. It doesn't matter whether it is horticultural crops or any other crop. It basically depends on the question we have. Like for forage crops, how we can quantify the post-harvest quality factors? Yeah, for forage crops, so for example, if we want to look at this in vitro digestibility, then it's the nurse machine which can help us to give us an estimate about that. I think you heard about this nurse presentation, and there you might have heard about all the details of this nurse machine. Okay. Can CT scan be used to detect storage pests? Also kindly explain the XRF in short. Okay, to the first question, yes, we can use CT machine to detect the storage pest. So it's basically, if you take a 2D image, we can look at the internal structure of the pest, the internal structure of the seed, and if the pest is there inside, it will be visible in the image. Also kindly explain XRF in short. Yeah, so XRF is basically used for estimating the elemental composition of this post-harvest products. So in our case, we usually use the grain samples to know what are the elemental composition, and most importantly, we look at this iron, zinc, and calcium content. And in the presentation, I have shown you a paper. If you go through it, you will get more details. Can we use XRF for other elements
other than FECA and ZN?
Yes, we can use. It all depends on having the calibration equation for that particular elements which you will be interested in. Can we detect seed-borne diseases or pathogenic spores present? Yes, it's possible. Can this technology CT be used in large-scale areas to identify poor seed filling patched in fields so that adequate measures? Yes, it can be used. And for your information, definitely we will be, the field samples really can go to CT, because manually testing all this will take a lot of time, but CT can make it very faster. Yes, definitely I can share the PPT.
Packaging materials also plays an important role in seed viability. Ma'am, please comment on this.
What do you mean by packaging materials? You mean the materials that will be used for? Yeah, it can, yes. How CT scan technology helps in seed production program. So in seed production program, so as I said in the presentation, we can use the CT technology for looking at the seed viability, and this will be extremely useful for the gene bank people to know whether the seeds are viable or not. How can I get certificate? I don't know what certificate you are asking.
ये सब हम बता देंगे सर्टिफिकेट कैसे क्या मिलेगा, आप प्लीज ये मैम से मत पूछिए. मैम आप नेक्स्ट क्वेश्चन पे आइए.
Can we join this harvest master beside the tractor? Exactly. So so the harvest master in general, it can be combined with the harvester machine. So with not with the tractor, but then the machine that will be used for harvesting the samples, and then you can get the real-time data. Which crop is best for forage? In my opinion, I think both the crop. And do we have any other questions?
I think अब कोई क्वेश्चन चैट बॉक्स में नहीं है मैम.
Yeah, okay, I'm getting another one. Can we use CT tech to determine the seed filling in field condition like a phenotyping sensor? Yes. So the CT machine right now, what we have is a benchtop, but then the later version will be the portable one, which will be used in the future.
Ankur, I would like to chip in a little bit for 2 minutes. Yes. Is that I'm audible?
Yes.
Yes.
Yeah, so some of the questions related to CT, whether it can be used, how it can be used in production technology, whether we can see the seed-borne diseases and all this. So if we understand the principle of CT, like principle of CT is image-based technology. So image, which is itself is a higher resolution, which goes up to the cell and molecular level. Then anything, so if you have insect can be detected, larva, egg can be detected. That's why we have initiated some joint program with plant quarantine unit, because whenever they have import or export, they have to do a proper testing whether there is any kind of seed infestation. So in that, it is very easy, like it's very non-destructive, and you scan and you will have the idea. Second thing, seed-borne diseases. So pathogen we cannot detect the pathogen, but if it causes any symptoms in that, which change the density of the seed, density in the particle density inside the seed, that can be possible to detect, but we have to take in CT, we have two models, 2D and 3D. 3D is really high, very high resolution. That kind of test can be taken. Again, in gene bank, they spend a lot of money for germination test, and then seed viability can be very good technology. Is a future technology. In seed production, sometimes breeder seeds are not enough, and they have to also, but they have to characterize it, like without shelling. They want to know, if they want to know shelling percentage. So in groundnut, not a problem, you sow the kernel, but in rice, if it is husked, it cannot be used for sowing. So without husking, you can have all these traits. So in seed production technology, hybrid seed technology, this can be used with a lot of ease. Like we scan the seed, we characterize them, we give the data, and we also return their seeds. So that is a proven technology which will not make breeders to do spend on the seed or on the time. So it is very fast, and we return the seeds, so they get their seeds. If it is less in amount, is quite fine. I think, yes, these are the two things I wanted to add in. Harvest Master, again, yes, with Harvester, it can be, it's a very portable. People can tie up in their bicycle also and take it to field. It's battery operated. It can be attached to thresher or harvester, and it can give the instant results. So that is also quite good technology for this.
Yeah, just to add a point of clarification regarding my previous answers to the question. So the pathogens, which are there are some questions related to visibility of pathogens inside the seed material, whether it can be visible through the CT scanning. So the microscopic pathogens might not be visible, but then if the damages that has happened through the pathogens, we can see that through the CT scan. Otherwise, if there is any larva sitting inside the seeds, this can be easily visualized through the CT scans. And we have some of the pictures related to that also. So the larvas can be made visible through that imaging.
Yeah, one of the questions from Dr. Tilok Nath, it's out of subject, but then I still like to answer whether sorghum or pearl millet both are fit for forage crops. So if you have to compare in terms of the nitrogen content or in biomass or digestibility. So in quality trait, digestibility is the one of the important trait. 1% difference in digestibility makes 6 to 8% difference in the animal productivity, like trout power, milking power, or body mass. So that is a very, very important trait. And if you see the sorghum, the digestibility percentage range, like something 50, or the best one goes up to 55, and there is a premium price for sorghum. But pearl millet digestibility is little less than that. So if you have a choice, sorghum will fit better than pearl millet. But in India, it's not distributed like that. It's like sometimes it's season wise. Farmer feed in Kharif pearl millet fodder, and then in Rabi sorghum, post-rainy sorghum, or in some areas sorghum is grown, some area pearl millet is grown. So it's accordingly.
So here is another question on the cost of Harvest Master. I think the Harvest Master which we have here is we bought a long back, so I'm not really sure about what is the current cost, but we can share the cost once we know through email to you if you are still interested to know about that. Okay, for the other two technology, also someone wants to know about the cost. Sunita, do you want to, can you please say the cost that is involved in these other two technologies, the CT and XRF?
So for us, like this, two instruments are of research grade, which is like, which usually fetch higher cost because it's a research grade, and it's utilized for various purpose. It's for multi-purpose. But if you go to the companies, nowadays they have a global research grade or the grade which is used for particular purpose. Like if there is a oil company, they want to only assess the oil. In NIRS, I'm saying in CT, if they are just interested for iron and zinc and all those. So CT is quite costly what we have, and it's a imported one, so it's something like of $50,000. But this is not the product by company, but it has been given by the Fraunhofer, which is a research organization of Germany. They have not released the final machine for commercial purpose, so that will be quite less. And XRF is quite less, is $10,000 of Oxford type. Bruker one will be little bit costlier than that.
Thank you.
I guess all the questions are over. Good evening, Tarunya ma'am and Sunita ma'am. Thank you for your inputs this evening. I'm Dr. Soni Gupta, joining from CSJMU Kanpur. Thank you for sharing your expertise with all the participants. And we had an experience of hearing a wonderful lecture from you. Thanks a lot. And for the participants, I'd like to share the information that we'll be meeting on Monday, because we are in a weekend mode. We will not be joining on Saturday and Sunday. So we'll meet again on Monday. Thanks a lot again, Dr. Tarunya. And thanks a lot, Dr. Sunita, from joining from ICRISAT. Thanks a lot. We'll be signing off now.
Thank you.
Thank you, Dr. Soni, and thank you.
Thanks a lot, ma'am.
And all the participants.
सभी पार्टिसिपेंट्स लीव कर सकते हैं।